Hey, I'm Britton Pack, a genetic counselor here, and I understand your 4-year-old son has been diagnosed with Fragile X syndrome, and it can be a very scary thing as a parent to know anything's wrong with your child. But I'm going to try to do my job to make this disorder make more sense to you two as parents, and that way you have a better understanding of it at a base level genetically before you go see the therapist, the psychologist, and pediatrician later this week to learn more about the disease. That way you go in with a base level of information that the rest of it can click with you. Fragile X is obviously a disorder, and it's an X-linked disorder, meaning that it is attached to the X chromosome, which is one of two sex chromosomes, both an X and a Y, which determines the sex of a human, as you both know. It causes intellectual disability along with a couple others, but it is one of the leading causes of intellectual problems in the nation. Since it's X-linked, it has different odds for each gender due to the differences in X chromosomes, with a male having one due to the X and the Y, and a female having two X's, with both receiving an X from the mother and the father. There's multiple aspects that factor into the disorder, but we'll start by looking into the X-linked aspect. Since females have two X's, this allows them to solely be carriers, or to have it also, so to be a carrier and not show it, or to have it and obviously display it. Whereas males, including your 4-year-old son, cannot be a carrier since they only have one X. They either have it or they don't. And if they do get on that X, they will exemplify the disease. Back to females. Since females have two X's, it means they always have to deactivate or inactivate one, which is to make up for a superior amount of X genes. Because if both X's were always used, there would be too high of a dosage of all the genes on the X chromosome, and this would not be good. So every cell in the body deactivates one of the X's, and the other one turns into what is called a bar body, which isn't important for what you're looking for is Fragile X. This body, it can affect how much it portrays in females. Since one is being inactivated, it can limit the dosage of Fragile X in the body. To start with the disease, it can start as a premutation, which we'll go into later when we get into other trinucleotide repeats and such. But it gets worse throughout generations, typically going from not having it at all to a premutation and to the full mutation, which is a sign of genetic anticipation, which is any time a disease actually increases in severity throughout generations. This actually means that your son received this from you, Mom, but that's not anything to worry about for you, and it's no reason to feel bad. However, don't use this as a reason to not have any more kids, because you're only a carrier, and that means that if you have another kid, it's 50-50 odds. So technically, your last kid obviously has it now, but your next one may not. So this is a 50% chance, since you have a healthy chromosome that could be passed on to your kid. Another thing that impacts it is males are more likely to have it since they only have one X. About 1 in 4,000 males have this to 1 in 8,000 females, so double the odds. Makes sense considering a female has half the chance, since they could also get a healthy X. The good news, if your son ends up reproducing and having kids, he won't be able to pass his mutation on, since males only pass on their Y to their kids. So this will not affect his kids. As the other professionals later this week are going to inform you, it is likely that your son will have greater symptoms, since he doesn't have another X to inactivate in other parts of the body. So this one X will be shown throughout the entire body. That's the main points of the X, so I'll get into some other things about the chromosome. A big part of Fragile X that we think is called fragile sites, and fragile sites are pretty important to Fragile X. A fragile site is a specific heritable point that tends to form a sort of gap, or so a breakage point or a constriction point on the chromosome, which is what happens in Fragile X. It happens in about 1 out of 20 cases. Also, among these fragile sites, there are about 7 that are folate sensitive, so it has to do with the concentration of folic acid. And these folate sensitive sites express trinucleotide repeats, which, as I said, we'll talk about later with its own topic. Folic acid is important in strengthening chromosomes, which is why we think this could be important. If there's a deficiency, it can lead to chromosomal instability. And this fragile site that we're talking about for FXS is called FRAXA, or FRAXA, and it's one of the 7 sites I just talked about. However, Fragile X isn't necessarily associated with the breaking of the chromosome, but more of the constriction. And at the very end, it almost looks like it gets constricted into a doorknob, which makes the X look weak. And that's kind of how Fragile X gets its name, because under a microscope, the chromosome looks weak in general. More research is actually being done between the correlation of folic acid concentration in this disorder, but as of now, we can't particularly say one thing or the other. So now we're going to talk about trinucleotide repeats, and as I said earlier, like a lot of the fragile sites have to do with this, and it's part of the genetic anticipation, as it typically, this is how it climbs from a pre-mutation up to a full mutation with the number of repeats that increase throughout generations. And just so you know, a trinucleotide repeat is a cluster of 3 nucleotides repeated in or near a gene. And for FXS, this is actually the nucleotide repeat CGG, or cytosine guanine guanine, which are part of the 4 bases, which are adenine, guanine, cytosine, and thymine. And then if you include RNA, we have uracil. Fragile X is caused by changes in the FMR1 gene, or Fragile X mental retardation gene, which produces FMRP, which is just the protein that comes from this gene, as all genes in the body produce proteins. This protein is actually a protein that is needed for brain development, and this is why mental disabilities come along with this disease or disorder. In Fragile X, FMR1 is unable to be transcribed due to the Fragile X and cannot produce the protein that is associated with this gene. As I said, the repeat of CGG is replicated many times due to what is known as replication slippage, which is just transcription error from the Fragile syndrome. So with genetic anticipation, in a normal gene, there's gonna be less than 44 repeats, possibly like way less. And when we start talking about the premutation is when you're in that area from around 55 to about 200 to 230. It kind of varies from person to person. A full mutation, though, is typically always seen as more than 230 of these CGG repeats. When there are more than these 230 repeats resulting in a full mutation, it causes a sort of a switch to flip in the gene, and it causes a chemical modification that actually inactivates the gene from producing the protein. This is what leads to our cognitive disabilities. These repeats actually occur in a region known as the promoter, which is a 5' UTR, or untranscribed region. So this area in DNA, it's not replicated, so it stays, and it's not going to be part of the gene that's transcribed or copied into the next sequence. Since the repeat is CGG, this leads to what is known as a CPG island, or any C or cytosine followed immediately by guanine, which is what causes it to be shut off and causes a hairpin sort of shape to be formed in the DNA, which I'll talk about in a second. Also in this, there are 3 initiator sequences, or INRs, that are upstream from the repeats that enhance transcription of the gene. Even though this transcription is occurring, however, translation is being inhibited by this hairpin. These repeats form what looks like a hairpin. It almost goes up and down on the DNA, and the DNA replicators can't get to it. This is what prevents the protein from being translated and produced. Also, amongst these CGGs, there are also a bunch of AGG inserts in between them. So even if transcription can occur, AGG acts as a sort of roadblock or speed bump to slow down translation. And it's just another obstacle that gets in the way, and it makes it too much for the cell to attempt to even transcribe and translate this gene and produce the protein. All the symptoms of Fragile X result from the lack of this protein. The lack of translation due to the repeats I just mentioned is known as a repeat-induced silencing, since the repeats are why the repeats can directly be associated with why the protein's not being produced. So now we're going to dive a little deeper into the protein known as FMRP, or Fragile X mental retardation protein, so that you better understand its importance in this disorder. FMRP is a selective RNA binding protein, which is very important in this case because it regulates the translation of mRNA in our neurons. It can inhibit translation by binding to the mRNA, or it can allow translation to occur by not binding and letting it freely go through the ribosome, as it acts as a sort of roadblock in the ribosome to stop the gene from being produced as smoothly. In FSX, there's a lack of translation repression, and this leads to a damaged synaptic plasticity, and a synapse is the area in a nerve where all sorts of neurotransmitters are being emitted and letting us know what's going on in our environment. Presynaptic damages can also occur in the neuron, such as functional abnormalities and an increased calcium influx to the synapse, which is not good, as calcium it sets a environment, and changing this environment in the whole body is not good. The human body is conditioned to have a set amount of calcium in this area that is accustomed in this situation. Due to these damages resulted in Fragile X, we can see that this protein produced by FMR1 keeps our neuronal activity at a normal human level, and this lack in normal activity is what is leading to our mental deficits that we see in this disease. Fragile X can also be attributed to epigenetic changes. Epigenetics is one of the more complicated topics in biology and genetics, and you might not have a great understanding of it because it's not talked about in most introductory biology courses if that's all you took. So let me give you a brief rundown. Epigenetics are changes in gene expression that don't change the genetic makeup. So it's sorts of things that are influenced by the environment, or things post-birth, post-fertilization, that aren't set, but they have an effect and don't change your DNA at all. So like all your CGGs stay the same. There's no changing of your genetic code. Your genetic code stays the same. Other factors are being added or subtracted from DNA and its environment to allow transcription to go as normal, to change the amount of transcription going through. Some of the common changes in epigenetics are methylation, for one, and that is by far the most common. There's chromatin changes, histone modifications, also known as I believe it's histone post-tail modifications, and there's also non-coding RNAs. The big one is definitely methylation. It's our most common epigenetic influence as a species. It's simply the addition of a methyl group, which is it's a carbon with 3 hydrogens, or CH3, and it just binds to another carbon. Also a big topic in chemistry, but it's not important in this case. It leads to a gene being underexpressed because these methyl groups compact the DNA and make it to where all of our RNA polymerases and other transcriptional factors cannot get to the DNA or easily access it. And this results from the CPG islands, or the CGG repeats in this case that we talked about earlier. And Fragile X, the embryo actually becomes hypermethylated, which means that too many methyl groups are being added to our DNA. And this occurs roughly around the 11-week mark of gestation in most cases, but it can obviously be less or more. And this leads to the underexpression of our gene and a lack of the protein that we talked about. Chromatin changes can affect it also, which are kind of what methylation leads to, but it can come from other things such as acetylation. Chromatin changes are just how compact our DNA is. So chromatin is what we view DNA as when it's in a cell. When it's tightly compacted, we call it euchromatin because it's all the same thing. And when it's put into this and it's all, or my apologies, heterochromatin is when it's tightly compacted and it's in a chromosome and it can't be accessed. So actually when our DNA is copied and translated, our DNA uncoils, and that's known as euchromatin because it's all the same, and you can't tell what goes with what and which came from which. So these chromatin changes either cause the DNA to tighten up and not be able to be accessed, or it loosens and it can be over-transcribed. Histones is another thing that can be changed, and histones are a sort of ball in the middle of your DNA that DNA just wraps around, and it wraps around it tight. You can add certain things to the histone. We can add proteins, add amino acids to it to lengthen it, shorten it, which can also cause changes. And also we have non-coding RNAs, which are things that can also affect translation. We have miRNAs and siRNAs, which both affect the amount of translation. With one degrading mRNA to where it can't be used, and the other preventing it from binding to a ribosome. A really good thing about looking back at epigenetics, though, is epigenetic changes may be reversible. Now, I'm not trying to get your hopes up, but if the Fragile X syndrome was caused by solely epigenetic changes, such as methylation, there's a chance it could be reversed. However, there's no guarantee, and there's not truly enough research into this yet. But with potential research, we believe that these kind of methylations and obviously other sorts of epigenetic changes can be sort of changed and reverted back and could reverse the damage of Fragile X. But since most of the times it occurs in the embryo stage, we don't know how this would affect, since we don't know if it would have a lasting effect since the body has never had this protein. But in review, the big things to take away are that Fragile X is a mental disorder which can cause a lot of mental deficits, but that it has a lot to do with sex chromosomes, mainly the X, but the Y causes it to be worse in men. To take away these fragile sites and kind of what it looks like. So the chromosomes being compacted at one end, and it makes our chromosome look fragile, and that's how we get our name. But the biggest thing is to take away the gene, FMR1, and the protein, FMRP, and how things such as trinucleotide repeats and the concentration of them affect this gene. But other than that, that's all I have today for you guys from a genetic basis of this disease, and I hope all goes well for you and your son, and I hope everything else you need can be figured out from our other professionals that you're going to see. And if you have any other questions, just feel free to reach out back to me, and I'll get you the information you need. And other than that, it's been a pleasure. Thank you guys for coming in. Again, my name is Braden Pack. It's been a pleasure talking to you guys today. Thank you very much.